Well, hello, and welcome to Bio 212 final exam review for A+P II. And the overarching question, of course, is how do I prepare for this test? I think what you need to do, bare minimum, is inside the back of your mind, have this idea that it's going to be 150 questions. Some will be true and false, some will be matching, and some will be sequence type questions. And over the course of the semester, I've tried to give you, obviously, examples of each of these as we're going along, with an eye to how you might actually see them over the course of the final exam, without feeling like I'm giving you the information on the final exam. There's always this sort of like, I need you to be prepared on your own because we're not going to be there when you need to access the information in the future. Thing that's always going on inside the back of, hopefully, every instructor's head, but not necessarily. You're going to be given 3 hours to complete this exam. That's essentially the span of time it takes to do the lab inside A+P II. So we'll start on like 5:30 and we'll go to 8:30. But beyond that, the most challenging question to answer is what is the most important information and what chapters are the most important. And what I would like to do is sort of similar to what I've been doing in class all semester long, is ask you to think about what it means to regulate normal homeostasis, right? And hopefully by this point in time, you have inside your mind's eye the fact that depending on the stage of life, either from fertilization all the way through to death, right, your body's constantly going through these changes that require some form of regulation. And when I say regulation inside A+P II, it not only means, do you understand that the autonomic nervous system is detecting all of these changes inside of your body, but that we're utilizing the endocrine system to actually affect what's actually happening out here inside the interstitial fluid of your body. Because remember, it's the interstitial fluid that actually has you live. And when it's out of its correct homeostatic range, bad things happen. So by way of example, let's get rid of the cartoon. Wham. Move on to the next one. Okay, in the course of like the next, 10, 11 slides, something like that, I want to walk you through how I would approach the review sheet that we gave you for the exam. And you're going to see here, what I've done is I've essentially cut and paste Chapter 18, Endocrine System, by way of example. And I'm going to ask you that same question again, which one are the most important hormones? And I'm also going to sort of remind you that we've spoken about this multiple times this semester, right? So at the top of your head should be ADH, antidiuretic hormone produced inside the hypothalamus, exocytosis, posterior pituitary gland of the hypophyseal portal system. It regulates normal water regulation. Okay. Well, then after that we have A+P, which is if you have too much water inside the body, osmotic centers inside the brain release BANP, right? But inside your heart, inside the right atria of your heart, if it gets stretched too much, it will release A+P to target the non-collection of sodium and chloride, which means water will stay to dilute sodium and chloride, which means your urine will increase, your urine output. And you can see what I'm doing here. I'm thinking to myself, if I'm a bag of salt water, what are the hormones that are going to regulate that bag of salt water? So hopefully that gets you to aldosterone. It's going to get you to angiotensin II, which is what A2 stands for. And you can see what I'm doing is I'm sort of creating this list of hormones. And the reason for that is I want you to think about their targets, cells. So for ADH, A+P, and aldosterone, the cell is principal cell. Principal cell. Cell, and it's not a pun either. It is the principal cell. We call it a principal cell because it principally regulates water levels inside your body. Notice when I started discussing these, I said the site of synthesis. Well, ADH, hypothalamus. A+P, I gave you 2 different locations. Aldosterone. Where is aldosterone made? Okay, you should know that it's inside the cortex of the adrenal glands, zona glomerulosa, right? A2, angiotensin II is converted from angiotensin I inside the lungs. So when we're talking about cytosynthesis, hopefully in that table you've put together all semester long, what you've done is you've collected that piece of information. This is not the entire list of important hormones. What I want you to realize, hopefully, is when I complete this down here, you're going through this, you're looking at it and going, okay, I know that for osteoclasts and osteoblasts, what hormones target it? Principal cell of the nephron. Well, guess what we just covered, right? Smooth muscle. Well, A+P I taught you that smooth muscle is regulated by epi and norepi. Right? But that's just the sympathetic nervous system, and therefore you have to think about what the parasympathetic nervous system is going to do there as well. And that means there has to be some form of a muscarinic or nicotinic receptor present there. Interstitial cells of the testis would refer to the reproductive system, specifically for male. Corpus luteum. Okay, where is the corpus luteum? What is it being targeted by? And if we're telling you none of these inside the review sheet, it means that we're probably going to give you some examples that are not going to have any of the hormones that will target any of these tissues. So we've got that list of 60-something hormones, right? We'll give you matching categories for the site of synthesis. B, and that's primary effects. What does it do? Okay, so these are tough, right? Which system, which tissue? I want you to continually think to yourself while you're studying, is this what I see in an adult? Is this what I see inside an infant? Is this what I see inside a pregnant individual? Okay. I will argue that if you had time to complete a Venn diagram of all of these, what you would discover is this is probably a more descript list of hormones you want to make sure you understand inside and out. All right? ADH, AMP, aldosterone, A2, insulin, glucagon, PTH, calcitonin, epinephrine, norepinephrine, and ACH. And notice that, again, I'm saying this in the sense that they're regulating the autonomic nervous system. Okay? Well, why? Well, the how and the why, it comes down to, I would argue that they support one idea. All cells need energy, and they need that glucose so that you can make ATP so they can do their specific job. How do we get each cell what it needs and when it needs it? We have to understand that if you are a bag of salt water, it's that interstitial fluid content that is regulated. Okay, now, this is almost too much in the way of sort of, hey, you should understand this by now, but if you don't, I'm hoping you're listening, and that you're listening to the fact that homeostasis is regulated at the interstitial fluid levels. And you have to think about what stage we're looking at. So when you're reading exam questions, you're thinking to yourself, are they giving me enough information to actually answer these questions? So let's go to chapter 26. This is the review material from your final exam, right? In the final exam review sheet. And how do you use this information? Well, inside chapter 26, we're going to have a matching category with a diagram of the nephron. I mean, essentially, you should be able to draw this at this point in time, but if we provide the diagram for you, you can pretty much guess we're going to ask you to identify what the individual areas are. And think about what we've done inside lab multiple times. I've had you draw this multiple times for a specific reason. So you know where filtration occurs. You know where absorption and reabsorption occurs. You know where the actual formation of the secreted materials is going to occur. And you notice it happens in multiple locations. Now, if I drew this out, it would take 2 seconds, and you would see that. What do we mean by different parts of the nephron? Well, if we're talking about the histology, where are the cells and where are, I should say, what are, which cells are where and that are functional? I should put an and in there, right? And that are functional. So some parts are not permeable to water. Some parts are permeable to water. Some parts we can regulate water levels in, right? What's the difference between aquaporin 1 and aquaporin 2? Okay. Which division of the nervous system promotes urination? Well, so far we've told you that SYMP can either ink or deke filtration, filtration. And if you look at the cartoon I've drawn for you several times, you're going to notice that is sympathetic innervation, SYMP, innervation at the afferent arteriole, right? Afferent arteriole, A-art, is what regulates that flow rate, okay? But you'll notice we have nothing about parasympathetic nervous system here, okay? So parasympathetic regulates urination, getting rid of the actual materials. And remember, urination, big fancy word for urination, the reflex itself is called micturation, okay? Mic. Sure. Right. What's the difference between a cortical nephron and a juxtamedullary nephron? If I were you, I would draw this out. But you're not necessarily me. Maybe you're the type that wants to make lists. And the list should actually have not just length, L-E-N-G-T-H, of nephron. I used to say loop of nephron. Loop of nephron. But what about vasculature? Okay. Sensitivity to hormones. And which ones? Put a question mark for that. And we could go on, but you know, ideally inside your head, you can see immediately how these things are similar and how they're different. What about just basic numbers? 95% of the nephrons inside of your body are cortical nephrons, right? Describe the process of urine formation. And initially, this isn't how it's written inside your review sheet. But if I'm you, I'm reading this and going, okay, I can make urine. Big fucking deal. Great. I'm happy for you. What you need to be able to do is say, I'm going to make dilute urine, or I'm going to make concentrated urine. What is the difference? Well, dilute urine is made in cortical nephrons. Concentrated urine is made in juxtamedullary nephrons. And then you go back and you go, oh, well, then I would have to use all of these things again, right? So that's how I want you to look at the review sheet. The review sheet is almost too simplistic. It's sort of, if you're not thinking actively about how to use the information, you're not going to, this review sheet's not going to be helpful to you. So I'm going to argue that if this is the respiration part, right, and so this is respiration here. Describe the cells found inside the alveoli in terms of their function. Well, remember, the alveoli are what type of cells? All right, they're epithelial cells. In fact, they're squamous epithelial cells. So type 1 and type 2, 1 is responsible for gas exchange and 2 is responsible for reducing surface tension, right? So if I'm you, I'm thinking, how did Mother Nature know where to put all of these things? Think about blood vessels for just a moment, right? Not that your lungs, at least here where gas exchange is occurring, are made up of blood vessels, but just adjacent to them are blood vessels. B-L-O-O-D-V-E-S-S-E-L-S, right? And those blood vessels are all unique, U-N-I-Q-U-E, to the responsibility that they have, responsibility that they actually have, right? So not only do we have 3 different types of capillary beds, but think about just the different types of blood vessels that you have. You have arteries, you have veins, you have capillaries, you have arterioles, you have venules. What are each of them doing? Do you know where you would find them? Who uses what? Do you remember that essentially an artery is different from a vein in the sense that the thickness of smooth muscle determines the total peripheral resistance? We'll come to that, or if we get to it, you know, when you actually get to the blood flow or conduit system that we know inside chapter 21. Okay, but the next part of this review is, when inhalation occurs, is the diaphragm contracting or relaxing? Well, if you haven't figured this out yet, I'm sorry. Okay? Diaphragm, activated by the phrenic nerve, contracts. What does it do? It opens up the area of the entire thoracic cavity, right? So I just answered both of these for you. Do you really think we're going to ask questions that simple? Okay? It's going to be, what type of contraction is going to happen? What are the muscles that are going to be used? You know, we want passive breathing versus active breathing. That's what I want you to be thinking about. Differentiate between the following nerves. The phrenic nerve is not a goddamn cranial nerve. It's not. Okay? The vagus and glossopharyngeal are cranial nerves, and they collect specific types of information, and they're going to innervate. Exactly what are they going to innervate? And does the vagus nerve innervate the lungs? Question mark, question mark? Or aren't the vagus nerve and the glossopharyngeal simply collecting types of information? Double check that. What percentage of carbon dioxide travels inside the plasma? I gave you the numbers here, and you don't even have to go look them up. 7, 23, and 7. And what's the percent that's bound to hemoglobin? Where and when? You know, inside the red blood cells at the external respiration point, namely the lungs, versus when we're at the internal respiration point, namely at the cells where we're going to send materials to the mitochondria. Notice what the question's asking. Okay, what percent is bound to hemoglobin? Well, 23% of it's bound to hemoglobin, right? 70% of it is bicarb, and 7% of it is soluble inside the blood. Okay? Another way to look at this is, hey, oxygen, O2, right? Whoops. Just turn it around so it looks like what percent of oxygen travels inside the plasma. Right? O2. So just because we're asking you about carbon dioxide doesn't mean you can't use it as a moment to think about what our oxygen is doing as well. But this is where I remind you, specifically, if we're talking about carbon dioxide here, you should know how it's traveling inside the plasma. It's bicarb, it's bound to hemoglobin, or it's dissolved directly into the plasma of blood. So what's the difference between internal respiration, okay, and say cellular respiration? What's actually happening at this chloride shift? And what's the Bohr effect? Remember, as the pH is dropping, the shape of hemoglobin is changing, and oxygen comes off of each of the binding sites just that much more rapidly. Okay? Does that make sense? Notice I put you here, internal respiration is at the capillary bed, not the lungs or the mitochondria. So just one more thing before I shut this off and then start again, because I don't want these things to go too long. Which areas of the brain are responsible for, which areas of the brain are responsible? Where do we go here? There we are. Sorry about that. Okay. So you should be thinking, okay, the pons and the medulla. Fantastic. But when do we use the pons? When do we use the medulla? Who collected what information? Namely, where did the glossopharyngeal cranial nerve send its information? Where did the medulla collect and send its information? And what type of information was it collecting and sending? Okay, so let's pause here and I'll start the next one. Hopefully you're getting an idea of how I want you to think about the review. Okay, and then we'll talk about just some general ideas as to what I think are going to help you be more successful. Okay, bye for now.